أهلا بيكم في دروس الأحياء لصف الثالث ثانوي لغات. Today's lecture we are going to solve the questions at the end of the chapter of immunity. So let's begin. The first question, choose the correct answer. From the biochemical means of immunity in plants is the biochemical, okay? So it's not tyloses, it's not gum formation or deposition, it's not cork formation. These all are structural changes. But phenol formation is a biochemical immunity. Okay, phenol formation. The lymphoid stem cells mature and differentiate into different types of T lymphocytes. Where does the T lymphocytes mature? In the thymus gland. Okay, the B lymphocytes are produced and matured in the bone marrow. The lymphocytes that found in the blood are B, yes, T, yes, natural killer cells, yes. All the previous lymphocytes which attack carcinogenic cells and transplanted organs, remember the cytotoxic T cells. تمام. It's not suppressor T cells. From the cells which have the ability to engulf all the microbes and foreign bodies, they all have the ability to engulf foreign bodies and microbes. So all the previous multinucleated white blood cells, macrophages, single nucleated white blood cells. Okay, question 2. Give the reason for thickening of the plant cell wall with cellulose and lignin. Well, thickening of the plant cell wall with cellulose and lignin is to make it difficult for penetration of the pathogens, especially after thickening by lignin. So to make it difficult to penetrate. Outgrowths extend from parenchyma cells neighboring the tracheids and enter through their pits when the vascular system is cut or invaded by pathogens. That's because to prevent the movement of pathogens to the other parts of the plants. It's like tyloses. Some plants secrete poisonous substances like phenols to kill pathogenic organisms such as bacteria or inhibit their growth. Okay. Thymosin hormone plays an important role in the action of the immune system because it stimulates the maturity of lymphoid stem cells to T cells and their differentiation into different types inside the thymus gland. The number of T suppressor cells increase after destruction of the pathogen. Remember, the T suppressor cell, it inhibits immunity. They bind to plasma cells, T helper cells, and T cytotoxic cells. This binding will help the T suppressor cells to secrete lymphokines which inhibit the immune response, where plasma cells will stop producing antibodies and many of the T helper cells and the activated cytotoxic T cells will die. Okay. The secretion of interferons increase in the blood infected with viruses. Because the interferons bind to healthy cells neighboring to the infected cells and induce them to produce enzymes and substances that inhibit the action of replication of enzymes of the virus. So that's how they combat the virus, thus preventing the virus from reproduction and spreading in the body. There are many types of antibodies. because each antibody has two identical antigen binding sites, and the shape of these sites is variable, different from an antibody to another. This leads to specific binding between an antigen and its specific antibody. So each type of antibodies has specific type of antigen. Tears and saliva are considered as kinds of natural immunity. They contain substances that lyses the microbes, so protect the eye from the microbes in case of tears, and in case of saliva it contains substances that kill microbes due to dissolved enzymes. Human is infected only once in the measles. We're talking about memory cells here, because after the first infection the body forms the memory B and T cells that can live for tens of years and may survive until death. These memory cells store information about the antigens. So if the same individual infected by the same disease again, the memory cells formed in the past respond to the pathogen once it enters the body, and they start dividing quickly to produce within short period of time, so that the disease doesn't show its symptoms. Okay, plants kill some of their infected tissues with microbes. Plants do that to prevent pathogen to spread to the surrounding tissues, so the plant can get rid of the pathogen by the death of the injured tissue. Question 3. What happens in each of the following cases? The entry of a microbe with a specific antigen on its surface into the human body. B lymphocytes recognize the antigen, which is specific to it, and attaches itself to the antigen by using the immune receptors present on its outer surface for starting the steps of humoral immunity to form antibodies at last and kill the microbe. A part of plant is cut. What happens if a part of plant is cut? The plant forms cork to prevent the entry of pathogens to the plant, and forms tyloses if the vascular system exposed to cut or to invasion of pathogens to obstruct the movement of pathogens to other parts of the plant. What happens if the plant is infected with poisonous bacteria? The plant produces some enzymes known as detoxifying enzymes, where these enzymes interact with the toxins produced by the pathogens and invalidate their toxicity. What happens when the deficiency of thymosin hormone in the human body? So there will be no maturation of T cells in the thymus. But if this happened, by the way, after a certain age, after the first 20 years, there will be nothing, because the thymus gland will be rudimentary, then it will be degenerated. Deficiency of interference in the cells, the infected cells with viruses. Deficiency of interference increase the reproduction of and spreading of virus in the healthy cells due to not inhibiting the replication of the nucleic acid of the virus. Okay, compare between natural and acquired immunity in human. Remember, natural immunity is the innate immunity. It comprises the barriers in the body. Acquired immunity involves the response done by humoral and cell-mediated immunity. Compare between structural and biochemical immunity in plants. Biochemical immunity involving, producing many chemicals such as phenol, for example, but the structural changes like tylosis and the changes happening in the tracheids and the injured areas of the plants, that involves the structure of the plant and not substances produced by the plant. Compare between B lymphocytes and T lymphocytes. Well, B lymphocytes, about 10% to 15% of lymphocytes, but T lymphocytes are 80% of lymphocytes. The B lymphocytes formed and complete their growth to become mature in the bone marrow, while T cells formed in the bone marrow and mature in the thymus gland. Types of B cells, one type, but types of T cells, three types: helper, cytotoxic, and suppressor T cells. Function of B lymphocytes: identify any microbes or foreign materials such as bacteria or viruses, then adhere to this foreign material and produce antibodies. But the functions of the T lymphocytes: helper T cells activate other types of T cells. Cytotoxic T cells, again, helper T cells activate other types of T cells and stimulate B cells to produce antibodies. Cytotoxic T cells attack tissues, transplanted organs, or carcinogenic cells. Suppressor T cells, this is that inhibit, these cells inhibit immunity to stop the immune response at a certain limit. Cytotoxic and suppressor T cells. Cytotoxic cells attacks, as we studied before, the foreign cells, kills carcinogenic cells and transplanted organs, while suppressor T cells regulate the degree of the immune response to required limit and discourage or inhibit the action of T cells and B cells after elimination of the pathogen. Okay, compare between chemokines and interleukins. Chemokines, they recruit, it's the message by which the tissue attract circulating phagocytic cells, which are found in the blood, and large number to the sites existing of microbes or foreign particles to prevent their reproduction and spreading. The interleukins, they mediate communication between different immune cells and between immune system and different body cells. Also, they help the immune system to perform its defense function. Compare between complement system and interference. The complement system, the complement attaches to antibody, as you know, and its different types of proteins and enzymes destroy microbes in the blood after their conjugation with antibodies, where they lyse the membranes of the antigens and dissolve their content. While interference, different types of proteins, not specific for certain virus, it's for all viruses, produced by T cells, large phagocytes, and cells invaded by viruses. The function of interference is that they bind to healthy cells neighboring to the infected cells and induce them to produce enzymes that inhibit the replication of the nucleic acids of the virus, thus preventing the virus from reproduction and spreading all over the body. Primary and secondary immune response. Primary immune response, it's the first response to infection for the first time, where the cells multiplicate and activation of humoral and cell-mediated immunity. But this takes a while, so the disease manifests its symptoms. It takes about 8 to 10 days. But the secondary immune response, there will be memory cells involved, memory B cells, memory T cells. So as at the time the pathogen enters the body, it will be destroyed by these memory cells, and it won't take time, so the disease will not be manifested. Okay, what's meant by biochemical immunity in plants? Chemical compounds resulted by the plants as response to an infection includes receptors, antimicrobial chemicals like phenols and glycosides, non-protein amino acids, and antimicrobial proteins. Tylosis. Remember, they close the roads carrying the pathogen. An overgrowth of the protoplast or the parenchymal cells which protrude into the xylem vessels and tracheids through pits when the vascular system exposes to cut or to invasion of pathogens, and they prevent the movement of pathogens to other parts of the plant. Like obstruction, زي ما كأنك بتسد طريق على حاجة. Lymph nodes, they are a long network of lymphatic vessels in all body parts, and their size ranging from pinhead to seed of small beans. Their function is to purify the lymph from any harmful substances or microbes, because the nodes store white blood cells that help the fighting against the disease. T lymphocytes, percentage is around 80% of lymphocytes. Their source in the bone marrow, but maturation in the thymus gland, and differentiate into several types like helper T cells, suppressor T cells, and cytotoxic T cells. These are the types of T lymphocytes. Macrophages, they include two types. The fixed macrophages, found in most body tissues, and their function are ready to engulf foreign particles as well as microorganisms. The mobile macrophages, they offer information which are collected about the microbes and foreign particles to the specialized immune cells found in lymph nodes. Okay, chemokines, they recruit the large circulating phagocytic cells which are found in the blood with large number to site of existence of microbes. Interference, again, different types of proteins not specific for viruses and produced by cells invaded by viruses. Their function is to bind to healthy cells neighboring the infected cells, provoking them to produce enzymes that inhibit the replication of the nucleic acid of the virus. Complement system, again, there are a number of proteins and enzymes. Their function that destroys the microbes in the blood after their conjugation with the antibodies. Inflammatory response, it's non-specific defense mechanism in the area of injury as a response to the damage of tissue caused by the injury of the infection. It involves swelling and redness and hotness of the area because of the increased permeability of the vessels, as you remember. Cell-mediated immunity, the immune response done by T lymphocytes, done by the receptors found on the membranes of T lymphocytes that give it a specific response. That's all for today's lecture. I hope you got the maximum benefit from it, and thank you very much for watching.